It's all yours, Indu.
Hey. Hello, Jeff. Thank you for joining us and addressing this forum here today. And we're really honored to have you. And I think for all of you, you probably who knows who Jeff is already. Jeff is an executive director at Packard Center for ALS research, and he spent most of his time on ALS research at Johns Hopkins. And also, I feel fortunate to have working with Jeff on Answer ALS Team, and he's also an executive director of Answer ALS research team, and also a professor of neurology at Johns Hopkins. And also he runs one of the country's largest clinic, ALS clinic. So with that, I will have Jeff take over the mic here and give us a presentation. And the first format is gonna be, we're gonna have Jeff talk about today's topic, personalization of ALS therapy and research. And after that, we will have a Q&A session. So feel free to write all your questions on the chat box here. And we will have Tara Tadimala and Sarah Diaz, two of our advocates here, who will actually be monitoring the questions and will ask on your behalf. So with that, I will hand over to Jeff.
Thanks. Thanks so much, Indu. Thank all of you for being here tonight. I talk to patients all the time. Never, let me just see what this number, never over 200 at once. So that's huge. So no doubt there's plenty of questions. And I think it's wonderful what Indu and all of you have put together by having such a big forum. Most of us that have ALS clinics do this locally with our local support groups, but nothing on this size. And I think the impact of having many patients and through Zoom, whether that's a benefit of COVID, I don't know, but it's certainly a benefit for many of us to be able to rapidly communicate both with patients and, of course, at the other end, academically at research. So again, thank you. And I know you're in multiple time zones, so I apologize. Here, it's just early evening. And I'm also a little wet. I just finished, I raise honeybees. It's a little side discussion we could have. And I just finished drawing frames. I have to do that this weekend. It gets a little hot and sweaty out here on the East Coast right now. So what I want to do tonight is cover a couple different things. We're fortunate that Ed Rapp last month spoke about Answer ALS. I'm going to dig a little more into that and give you background. But I thought I would take advantage of seeing all of you and go back a step and tell you a little bit about ALS history. And I don't mean ancestral history, but what's the modernization, if you will, of ALS trials. Because most of you are here, you don't wear deep science hats. You hear about what are we doing to eventually find therapies for you, which has been the core of my career. So as a quick background, so as Indu said, I'm a neurologist. I trained at Johns Hopkins. I've been here in Baltimore 30 plus years. I'm an East Coaster, so I apologize to those of you who anywhere else in the country, you're missing out on the most important part of the country, East Coast. I run two different things. I have both a PhD and an MD, so my whole career has really been devoted to understanding the basis of ALS, using that understanding to really help develop therapies, whether I do it or that science is used by drug companies or other academics. The whole mission is really that bridge of basic science to clinical science. And I'll go through a little bit of the history of that in just a second, not me, but in general, how we've done this. So that's actually why on one end I founded and run the ALS clinic at Hopkins and Packard, which is Packard Center. It's actually not a clinical center per se. It's a research center. It's organizing researchers to collaboratively work together. And if I have time, I'll tell you more about it. But I'm going to pull up some slides because, by the way, a couple things. I apologize. I talk coast. I favor the East Coast because I grew up in New Jersey. And we all talk fast from New Jersey. So I apologize for all of that. And
thank you, Jeff. If you hadn't said that, I would wonder when it's going to happen. So,
so I always sort of throw that in. We all have biases. I'm sure all of you have biases. I have my own biases. One other thing I'll tell you right up front. As a clinician, I'm nothing but blunt. And I don't mean that in a mean way. I wear a science hat strongly, and I deeply believe that patients need to be informed. Not in a harsh way, but they need to know what the real science is, because in every disease, as a neurologist, I see many different diseases, although I focus on ALS, but I'm exposed to many diseases. And this community, over the decades I've been dealing with this, is faced with a lot of excitement, also a lot of hype. And I refuse to hype. as a scientist. You need to know the facts as they are. So you're going to hear me be blunt throughout this. And I'm going to apologize that I've offended some of you, but that's just the way I operate. You need to know what the real facts are as I see them. And to that end, I'm going to switch and I'm going to share, share screen, and you're going to see in a minute slides. And by the way, this is, although I am, oops, now let's get to PowerPoint and see all my emails. All right. So although this is going to focus about Answer ALS largely, because that is about the personalization of ALS therapies, and that comes about because of the great failures, quite frankly, of decades of ALS research and, more importantly, clinical trials. So it's important to have a sort of a basis for why we do things today, how we got to there, because many of you are brand new to this community. You may have had the disease for 6 months, 6 days, or 6 years, or even decades, but I've been doing this longer than all of you, I'm pretty sure. And I need you to understand how we got to where we are today, because it just gives you a perspective. So really ALS trials, and I'm going to focus mostly on trials, not the science. There's a lot of science behind this. And it reminds me, I left out a slide, and I may try to pull it up. Really so these are what might be called epochs or advances in ALS clinical trials and research, really. So from the decade of 85 to 95, which is very early in my career, things first began with what are called anecdotal trials. You know, and the one of them that comes to mind was a researcher out of Canada who said, you know, I hear calcium's important in the nervous system. Let me give a drug we use for calcium for the heart. Let me see what it does in ALS patients. I'm going to try it in 10 patients. And these are real published studies. And the drug failed. Oh, that meant calcium's not important. Well, quite frankly, an N of 10, we now know, okay, that's junk. Frankly, that's pure junk clinical trials. And when these trials were done way back then, and this is just one of many, the researcher didn't know, well, did the drug actually get into the brain? Because ALS is a disease of the brain and spinal cord, not your blood, not your heart, not your liver. The drug's got to get to where the disease is. And so many early trials were carried out with not a clue if the drug actually got into the brain. So it's sort of like throwing, I don't know, a drug on your skin, and most of you would know, well, okay, a drug on my skin's not going to get into my body. And that's sort of the equivalent. By the way, I like to use a lot of analogies. And so, but that ALS began with these, quite frankly, in today's world, just crappy anecdotal trials. They were valiant attempts, but not really well designed. And the real modernization of ALS trials began in the early 1990s because laboratory research had taught us that when you take rat and mouse motor neurons, remember ALS is a disease of spinal motor neurons, and I'm not going to give you a whole background on ALS. I'm going to hope you're mostly familiar with that. I'm happy to teach you that. And cortical motor neurons. These are very specialized cells in your nervous system. Aren't many of them in the grand scheme of your whole brain, but we had learned if you take those cells from embryonic mice and rats, they'll very quickly die. But if you give them things called growth factors, like hormones, kind of like insulin, but not insulin, and a variety of these have been described in the literature, in very good literature, that these would keep motor neurons alive. They'd make them grow back again in cultures of these, not human, because we didn't do human cells way back then. And there were a whole bunch of these, and they were remarkably effective in vitro, meaning in these culture dishes. And that led to the first set of real industry trials. Regeneron, in fact, now you hear a lot about Regeneron because of COVID. Quite frankly, a very good biotech trial. It was the very earliest days of Regeneron. They did CNTF, what's called ciliary neurotrophic factor. By the way, these letters all mean different things. GDNF, another one, growth-derived neurotrophic factor. BDNF, IGF-1. These were all tried in trials. And by then, we actually started making better trials, not an N of 10, maybe 100, maybe 200 patients. And in those early days, trials were all about, did someone live longer? And we'll get to how trials have changed, but ALS being, quite frankly and honestly, a fatal disease, we looked at it, we as the whole community, like cancer trials, which cancer trials are usually about how long does someone live and can you extend the life of someone. So those early trials were quite long. They were a year or even 2 years in duration. Some of the IGF-1 trials, one of those listed, was a 2-year trial. Half the patients were on placebo for 2 years. We don't do that today, but that's how things began. They all failed. Obviously, they failed. By the way, there's going to be a long list of failures, so they all failed. But we began to learn about how to do modern trials. However, and many companies were engaged in this, pretty good biotech companies in those early days. Not all of them, but mostly. You're going to hear me be critical of companies along the way, because I am. But it got the academic community and the companies engaged in trials. But because there's repeated failures of these really big trials for ALS, biggest ever after, essentially since ALS was discovered, companies dropped off the map. They stopped doing trials in ALS. But except for one, by 1995, riluzole had proven to be effective. Now, I was lucky. I was a junior investigator then. I was the local PI, that's a principal investigator. There were 5 sites in the United States and a whole bunch of sites in Europe that ran that trial. That was a survival trial as well. Many of you know about the measures that we use in ALS called ALS Functional Rating Scale. That didn't exist back in the 90s. Again, it's important for history to know these things. Those days, those trials were driven by European investigators, and they deeply believed the only way you would learn about ALS is to do a survival trial. Today, we don't believe that anymore, but that's how science evolved. And riluzole was proven to be effective. Now, we all know it's by no means a major blockbuster, but actually after 3 decades or close to 3 decades, it remains the repeatedly effective drug. By the way, I don't have stock in the company. I don't own anything. It's just true, because every big trial since then, every big pharma trial since that original riluzole trial has used riluzole, and it always works, albeit in a tiny way, a way that we didn't want. But you have to understand, in 1995, no drug worked in Alzheimer's, ALS, Parkinson's, any of these diseases. This was the first drug that actually changed the course of neurodegeneration in a neurodegenerative disease. That's the class of Alzheimer's and Parkinson's. So that was actually pretty exciting, in spite of the fact that it was a small effect. Now, again, to keep in mind before I go on, we knew as physicians that small effects eventually lead to big effects. I don't mean with the same drug, but the first effects in cancer with lymphomas was, yeah, you're going to die, but you lived 3 or 4 months longer. Oh my God, that was fantastic. Of course, years later, we learned that when we added multiple drugs in, cancers would be 3 months, and 6 months, 2 years, 5 years. But that was the starting point. In fact, in those early days after riluzole, we would meet with a lot of cancer researchers to get an idea, how do we build on that? What do we do next? And the downside of the original riluzole trials, because it was such a small effect, everyone said, it's a shitty drug. I apologize. I hope there are no kids on the phone because I swear a lot. I am from New Jersey. The fact is that we moved on to other drugs. We actually never went on to really beat the heck out of the drug to see if it could do more out of it, if a higher dose would even work more. We left it. And then what happened is that was the end of pharma's involvement. First drug approved, all the growth factors failed. And then what happened in the next decade was the growth of academic trials. And this was the growth of a program run by myself, Bob Brown, then at Mass General, Merit Sokovich, then very young, quite frankly, a colleague of mine, Jeremy Scheffner, and we formed something called NEALS. Actually, NEALS is Northeast ALS. You might say, wait a minute, how is this a national-wide group? Because way back then, academics has politics, just like the rest of the world, and there were actually different groups. There was it still exists, the West Coast ALS Research Group. There was the Northeast. There was the middle of the country. And these fractions were trying to start their own trials. These are the era of academically driven trials, ideas that came from us as researchers, because companies weren't coming to us with their drugs. So we were doing our own research and saying, maybe this drug could work. Maybe minocycline could work. Maybe topiramate. Drugs are already available based on our preclinical research. And NEALS was one of those groups. And today it is now the dominant academic group. But back then it was just a starter group. There were many of them. And we began to optimize. What's the better way to do a trial? And really in that decade and a little beyond that, we optimized and changed trial design. Now keep in mind, trials are really important, how we run them. But behind that, it's really the drug that's most important. You can, I was trying to think of an analogy. If trials are like a motorized vehicle, you can ride a little go-kart, that's a motorized vehicle, meaning what's the drug, what's the kind of vehicle, or you can drive a Cadillac, a BMW, you name a fancier car. So they all are a structure, and trial design improved the structure of the trial. They had nothing to do with what the drug was. And even today, we're going to talk about trials like platform trials. These are great designs, but a great design with quite frankly, a shitty drug is still, in the end, crap. It always depends. The key thing is, what is the drug? Now, given trial designs certainly provide greater opportunities for patients, Merit's talked to you about how important that is, but opportunities for a crappy drug is still crap in my mind. That's where I wear the science at. Because at the end of the day, I don't care how good the trial design is, if it's not a good drug, you gain nothing from it. You've got to start with a good drug, but you want to design today that's better treating patients. So I told you in the beginning, we had trials where half the patients were on placebo. There was no open label back then. Things have changed, and that's very good for patients. But what has to really change are what are the drugs? So I'll get to that in just a second. By the way, I guess you're muted. Feel free for someone to step in and stop me. So what happened was really in those decades, 95, 2000, early 2010, 12 or so, mostly academic trials dominated the landscape. Creatine, topiramate, drug after drug, various antioxidants. Occasionally there were biotech companies involved, little tiny ones, not the big pharma companies, and they were almost always poorly designed trials. They were looking to make a name for themselves and get money. Again, I'm being a little harsh, but it's a fact. What happened was, from 2010 to 2015, there was a resurgence of now interest from biotech and started to get interest in pharma. At first, these were new small molecules. Small molecules is the term we use for drugs like, I'll say aspirin, chemicals. And stem cells grew in interest because from 2000 to 2010, we were growing our ability to study stem cells. So there was a resurgence of those kind of one-off things, biotech companies. And also with all these little trials, you saw more and more hyping by companies. By the way, you'll almost never see hyping by big pharma. Biogen just published a major paper. We'll get to that at the end of this talk. You don't see hyping by them because they know they're not supposed to do that kind of stuff. And they have the funds to do things. And so we have often different, well, not everyone does, but I certainly reflect, we have different respect for the way a company approaches us with trials. Fortunately, many, but not all companies come to us for trial design today. Not all, some insist on doing it their own. Some insist on whatever we're telling them, doing whatever they want, bad idea. But fortunately, because of Niels and other groups having a better idea of how trials should be done, there's a gelling of how we do things better. And that reflects on the other column of this. There's an improvement in trial design. We no longer do, for the most part, survival studies. We have now ALS FRS. We think we can even do trials instead of a year, maybe even as short as 6 months. We haven't fully validated that yet, but that's good. That's certainly better for patients, but it's not better if we don't really find drugs that work. And then slowly there was a huge interest now, a resurgence of big pharma. That's important because big pharma are the ones who can carry out well-studied drugs with a lot of science behind them. And this jump in part reflects the fact that in 2011, the new gene that was discovered in ALS, called C9orf72, we call it C9 for short, because that drug accounts for up to 10% of sporadic ALS patients who have no family history. And in fact, in some countries in Europe, it can be as high as 20% of apparently sporadic patients having that gene. And it's a huge fraction of patients with inherited ALS, and it's common in certain dimensions. So suddenly you have a well-defined starting point for the disease with a very rapidly evolving biology around that mutation, gaining interest in companies. That's actually important. Whether my patients have C9 or not, getting more companies engaged is to our benefit as a community. We need them to carry out, to get interested in ALS and begin to spread their wings to sporadic as well. So from 2015, now we have all changes in ALS. Obviously, you've heard already from Merit about the trial design, the platform trial. We've also been understanding biomarkers. So if the drug's working or not, we want to know early. So we're beginning to have a sense of what are the markers in the course of a trial that tell us if the drug might be working. Now, we haven't had that yet. We have the markers. We don't have a proof that the markers work. I want to be clear about that. The only marker that we routinely are using now, I say routinely, we're talking about the last year or two, is something called neurofilament. I think you've heard about neurofilament. It's a protein in the long nerves, the axons, and it leaks out when neurons are damaged. By the way, it's not unique to ALS. Every degenerative disease has this leakage of neurofilament, strokes, epilepsy, multiple sclerosis. So by no means think it's unique to ALS, but it's a marker of injury. And we actually know that it's an important marker of injury because in spinal muscular atrophy, the baby version of an ALS-like disease, a disease that affects infants, where infants, when they have the most severe form, die within weeks to a month, we know the most powerful drug today in any degenerative disease is nusinersen, which is the drug that fixes that SMA, spinal muscular atrophy defect. And those babies who have very high levels, that drug not only stops their disease almost completely, but that neurofilament protein drops. That teaches us that that reporter, that biomarker, could be a very useful tool as we evaluate drugs in ALS. And then the other thing that's also novel to ALS, and I'll talk a little bit about this in just a minute, are of course the apps. How do we try to learn more about patients without them having to go from, say, Kentucky to Baltimore or wherever, when they live far from centers? We miss out on the opportunity to really bring patients into trials to understand their disease, because it's just difficult as the disease progresses to actually follow them. It's hard for them to get to trials. So the advent of both telemedicine, which is great, though has its limitations for really evaluating people through a screen, but these apps that were being developed for an iPhone or an Android really increase our ability to understand what's happening to you without coming to see us. By the way, it's still important to see clinics if you can, just because there's many other people that help you. Again, the other major interest is a huge growth in interest by biopharma. We now are beginning to focus, and this is where we're going to take off in just a minute, talking about subgroups. ALS patients may look alike when I see them, even though there's different courses, but I know if I see a patient who's fastly progressive, that could be SOD1 mutation, or it could just be that they're sporadic. I can't look at a patient and know that they have an SOD1 mutation or C9. There's absolutely nothing clinically that teaches me that. So clinical observation, it's old style neurology. Modern neurology, you've got to understand the molecular biology that underlies the disease. And now we know that there are many new targets from studying real patients by tissue that teaches us about the disease, and are getting to subgroups. So I wanted to do this, actually it wasn't very fast, but you need to sort of have, I think you need to have the sense of what's happened over all the years. What are we doing now that's different than before? And why have we gotten to this point? So I'm going to move on to now telling you, go back one step. So most of you know, of course, that ALS is mostly sporadic. It happens out of the blue, 90%. And a small percentage, about 10%, is inherited in families. And early on, we learned about the first gene in ALS, SOD1. It's only now, we now know, found in a very small percentage of patients. Only about 10% or less of familial patients have the SOD1 gene. The most patients with inherited ALS have the C9 gene. And then all these little circles, there are lots of other genes that we've discovered, not me, other colleagues, that cause very rare forms of ALS. That doesn't mean they're not important, but they're really rare forms. We as a community have been driven scientifically to study SOD1, because you can see it's the first gene was discovered by Bob Brown and Tipu Sadiq way back in the early 90s. And for literally almost 2 decades, we spent most of our research, preclinical research, trying to understand that. Turns out a lot of what we learned from SOD1 is interesting, may have very little relevance to most of sporadic ALS. Now, being general here doesn't mean we haven't developed great tools, but much of what we've done around SOD1 was we built a mouse that looked just like ALS, well, sort of looked like ALS. We used that mouse to study many drugs. And let's face it, that was successful. I'd be sitting here with a large medicine cabinet full of drugs, but we haven't. We failed finding drugs using those tools. And I'll tell you about the platform that's changed. But I do want to point out, again, I didn't put a lot of slides. Well, actually, I did put a lot of slides, but I didn't put all the slides in. One of the things you as a community must understand is, I'm guessing many of you, I hope all of you know what a phase 1, a phase 2, and a phase 3 trial is. Phase 1 is when we have a brand new drug, never been a man or woman. We learn about it very quickly. Does it hurt your heart? Do you pee it out? How do you get rid of it from your body? Phase 2 is when we begin to study a drug to see if it has efficacy. And by all accounts, by FDA and by good science, you need to prove twice that that drug works. Because a trial of 50 or 60 patients in a phase 2 is interesting, but here's the hard facts. In almost all of medicine, and certainly in neurology, 80 to 90% of drugs that are positive in a phase 2 trial, when we try them again in a phase 3 trial, fail. So this gets into the hype. Just because a trial looks excited in a phase 2 trial, you see that all the time in the newspaper, unfortunately does not mean it's really going to work in all patients. And you need to do that experiment twice. It's just a fact. And that's repeated over and over and over again in medicine, that high failure rate. It may be, in fact, because we're treating everyone, when in fact with that drug, we should only be treating a certain subset of patients. And this gets into the next slide. The fact is, without personalized medicine, we do this in ALS and Alzheimer's and in many diseases, we lump everyone together. So if this picture is a picture of ALS or even breast cancer, everyone used to get lumped together. And in the end, there was often no benefit. Maybe even some people did poorly. Maybe there was a benefit, but only 2 people out of 100 show benefits. You don't know if it was real or not. There's no way to really ascertain if that's true. But if you understand the mechanism of disease, you begin to target the right drug to the right patient. This is exceptionally well known in cancer. So when I trained in medical school, you look under the microscope, and you'd say, oh yeah, it's a breast biopsy or prostate biopsy for cancer. And you'd say, oh, those are cancer cells. They're dark, the nucleus is dark, they have a certain look to them. And then we would treat everyone with the same drug. But what we learned years later is if you take those cancers and now you analyze them for the molecular, the cell biology, the biology of those cells, all those breast cancers are not alike. There are different molecular biological reasons why some breast cancers are different than others. You can't tell that simply by looking under the microscope. And from that kind of analysis, we began to subgroup and began to treat patients like with cancers. And that's been the greatest advances have occurred with personalization of cancers. I know very well that not all cancers can be personalized. We've been very successful as a community saying, oh, you've got melanoma. You've got a melanoma that responds just to this kind of cancer. And a great example of that is, some of you may or not recall that Jimmy Carter, our ex-president, had a melanoma that had spread to the brain. As a neurologist, I know when melanoma goes to the brain, that's it. You're going to die. It's going to bleed. It's horrible. And yet, because of targeted therapy, those tumors melted away in his brain because of that kind of personalization. So enormous advances in cancer. Now, why were we able to do that in cancer? I said it earlier, you can biopsy the cancer. Oh, shoot, I can biopsy your skin, your liver, your heart, your pancreas, your bone, you name it. I cannot biopsy the brain or spinal cord, because if I take a chunk of your spinal cord out, you're permanently paralyzed and probably have what's called a permanent central pain that we can't stop. Your spinal cord is the width of my index finger. About 2.5 feet long or so. Imagine sticking a needle half the size of a garden hose into my finger, pulling out a chunk of tissue to your spinal cord. That's it. It never grows back. Human beings, rats, mice, monkeys, dogs, cats, we don't regenerate the nervous system. Maybe salamanders do. We're not salamanders. And so we don't biopsy except for when it's a brain tumor, then you're taking tumor tissue out. We can't biopsy the nervous system. So the very advances made in cancers cannot be done for neurologic diseases like ALS. That's until about 10 years ago, when we started to learn how to make stem cells. So there's a great controversy in the past about stem cells that come from women's eggs. We don't do that anymore. We know that we can take your blood cells or your skin cells, a little tiny biopsy of your skin or just a sample of blood from you, and we can reprogram it. We can use molecular tools to turn that blood cell back into a stem cell, which we can then turn into your motor neurons. And that's called induced pluripotent cells, or IPS cells. This is a revolution in medicine. It's changed everything. We can almost make every cell in the body. Now, some people think that's important for using them as a cell therapy. Yeah, probably not yet. That's probably mostly science fiction or hype. But by having a biopsy, I am now on equal footing with what we do in cancer. And if I could biopsy every single ALS patient, could I begin to understand what are the differences among those patients? And by having that biopsy, having those living cells in culture, I can, one, figure out, are there different kinds of ALS? And because they're living cells, I can find out what is the pathway, what is the molecular events that make one patient different from the next. And again, having those living cells, I can begin to test drugs in those cells before I test them in the patients. And finally, because I have those cells, I can learn about the differences. What are those biomarkers that allows me to separate patient 1 from patient 2, because those are their living brain cells. I can use that information in a proactive way to say, let's test their blood, or do a spinal test and set their CSF to say, do they have those biomarkers? Or are they in fact in group 1, group 2, group 3? So I very quickly described what is Answer ALS. And I know Ed last month reviewed some of this. I'm going to re-review it. Nothing beats repetition. So Answer ALS is a program looking at 1,000 patients around the country, 8 sites around the country, Johns Hopkins, Mass General, Ohio State, Emory. I'll go through this real quickly in just a second. Every patient is followed for years. So it's not a single visit. We want to know the different courses of patients, how patients progress slowly, fast. Do they begin with problems in their speech? Do they begin with problems in their arms? We have all of that data over at least a year. So we have what's called deep clinical data. And just to be sure that we get even more data, because patients, you'll see in a minute, not everyone can come back to clinic. We developed an app. So when they're not in clinic, they can use their Android or iPhone to collect even more information about them, their movements. And I'll tell you about the app in just a second. The blood that we collect gets sent, some of it gets sent to Neurogenome Center, and the entire genome of that patient is collected. Some of it gets sent to Cedars-Sinai, as you'll see in a minute. This is a huge collaborative network, because everything that I've done for my entire life has been about collaboration. No one single physician or scientist can figure it all out. They do that, they're egocentric, and they're making huge mistakes. Cedars-Sinai makes those iPS cells, makes those motor neurons, and now that we have the biopsy, a true biopsy, it gets analyzed by every biological parameter possible. I'll go through that in a minute. It generates huge amounts of data. And then the idea is to take all of that data, integrate the clinical data, all the biological data, and begin to find out, are there subgroups? I'm going to walk you through that in a second. This is a boring slide. This says this has a real structure. I formed this group about 2015 with the great help of Team Gleason. And then we had multiple people get involved, and we met Ed Rapp last month. Ed is the chairman of our advisory board. Multiple other patient representatives are part of our board. And then we have a whole team of researchers representing the stem cell group, the group that do the biological analysis, the clinical group, all of whom work together. We have somewhere between 2 to 3 Zoom calls every single week among different elements of this group to keep us working together as a team. We're an academic factory, if you will. And there's huge amounts of data flow. There's fluids that have to be moved from one site to the next. There's repositories of data. There's cell lines that have to be kept. And I'm going to go through a little bit of that to tell you the complexity of all of this. And this is actually overseen by a colleague of mine at Johns Hopkins, Emily Baxi, who's really the taskmaster that keeps all of the gears greased and working properly to make this move forward. So here's an example of the kind of things that we do. So the answer, I'm sorry, this actually was the wrong slide to show, but Answer ALS is now fully enrolled. We enrolled 1,000 patients in just about 3 years. That's about 10 times faster for the number of patients in any clinical trial ever in ALS. That's important because we needed those patients in the trial very quickly. The sites are shown here. As you can see, we're a little East Coast centric. I told you I'm from New Jersey. But in reality, we chose clinics that we knew a lot about, and we needed to enroll patients fast. One of the reasons clinical trials are expensive, there's a lot of expenses just running the clinics, collecting information from patients, seeing patients. And the sooner we can do all of that, the lower our costs were, because the team that, and you again met Ed Rapp last time, Ed's was, as you probably know, was a CEO at Caterpillar. He knows how to run a business, and he wanted us to run not like pokey academics, but very rapidly. So this team was built to run fast, and that's why we were able to enroll so fast. And these are the various PIs in each of the centers that sort of oversaw their clinical activities in each of the different centers. And then we followed patients, as you saw. We followed them every several months. And not uncommon to any ALS trial, here at, let's just say here at Hopkins, we enrolled 271 patients initially, but over time, less patients came to the clinic. It's not because they hate us. I hope they don't hate us. It's because it just becomes difficult to follow patients sometimes. They're ill, they can't make it there. Maybe they are coming from Tennessee. They're not just in our backyard in Baltimore. And as you can see across the board, that's very common that you lose patients. This is the case for all trials almost. You lose patients over time, whether it's because of their illness, they passed away, or it's just challenging to get to a clinic. And this is where, if I could show an offset, these were apps increase, even though we've lost patients, we increase the data over time because of those apps allowing us to collect data. Now, importantly, during the course of the trial, we also collected entire genome for every single patient. We have their entire genome. And that was in part to discover initially what genes they have, other ALS genes. But as it turns out, when you do an entire genome, you actually learn about all of the other genes that they might have that may be causing other diseases. Acute cardiac death genes. And there are 57, 59 different genes that the American Academy of Genetics mandates, or at least recommends, that if you find that gene in a patient, or whatever else you're doing, you should be telling that patient. Because although my ALS patient may have ALS, they also may be carrying in their family a sudden death onset gene from cardiac problems. They need to know about that so they can determine if they want to tell other family members. So in this program, we actually built into it, as we began, an entire genetics consortium, and I'll come back to that in a second. This is the kind of data that we follow. Today, as I mentioned, we don't do survival studies. We do ALS functional rating scale. And each of these colored lines are different patients that we follow. Some patients, as you can see in the slide on the right, progress very rapidly, and some progress very slowly. And you might be able to begin to think, well, maybe these are different subgroups. They might be. It's a clinical progression. Is there biological underpinning? This is a crude assessment. One of our team members, Ernest Frankel at MIT, Massachusetts Institute of Technology, as an incredibly talented graduate student, Divya, who's begun to take this data, in fact, this data, as well as all prior ALS trials, and use very elegant algorithms to say, well, are there actually different courses for ALS patients? We often say fast versus slow, but actually, Divya can find about 5 different curves, and we break this down, these colored curves. Some patients are very, very slow throughout their course, while others look slow, but then really pick up speed. And this is important for us because I've trained all the years I've been doing ALS that most patients follow what we call a linear course. Whatever they have, they follow in a linear course, but these are not all linear. Some patients look linear, and this is where knowing this data is important, because if a little tiny biotech company says, oh my God, look, I studied my patients for these 4 months, and look at that, I project that they're gonna look like this, and my drug made them look like this line. Well, actually, this group of patients were going to drop much faster. So looking at only that short period of time does not really teach you the trajectory of their disease, which is why sometimes you really do need longer timelines to know, is a drug working, versus an artificial extrapolation of this line out like that. So bits, and I'm not going to go through all of this data, but I'm going to give you bits and pieces. So that's part of it. We begin to analyze these subgroups of patients. And the other part had to do with this app. For years, we would see ALS patients, but we'd never have any follow through. So my team at Johns Hopkins, who are actually ex-Microsoft programmers, developed an ingenious app where we could follow multiple different elements of a typical disease in ALS patients. And these are actually, we had artists that drew all of this, animated these cartoons in the app that we developed. And the team, again, not me, this team, and there were motor functions. So you can't do this with your toes, but you certainly can do it with your hands. You can trace things, and we can time how accurate you are with tracing, how fast you are with tracing. And then probably the most creative and novel parts of this, we have what we're called bulbar functions, speech functions. And the app will show you sentences that you have to repeat, 10 different sentences. And we save those sentences for an analysis. Or the app will have cartoons that you'll look at and you'll describe what you see, the words that you use. And the critical thing is we have a really wonderful partnership with IBM Watson, a partnership that actually Ed Rapp, arranged for us. Watson takes this data, and we actually now know that just looking at speech, the words used in speech and the way they use the phonation allow us to project the course of an ALS patient better than my clinical exam, better than ALS functional, or equal to an ALS functional rating scale. And in addition, by analyzing the words used, we can detect very early cognitive changes in patients. Because honestly, we know, although we didn't know before, that some ALS patients do have cognitive problems. Their algorithms can detect that just by the kinds of words they use. These are really powerful tools that we'll be able to expand way beyond Answer ALS one day. This program started with the local team at Hopkins, and now Microsoft is partnering with us to help modify this app as well. And this app is now what's known as open source. Anyone can download the program and work with it. I mentioned before that we now also give out genetic information. Even the genes that we've learned within our program have been shared with researchers around the country. And in fact, the new ALS gene was discovered because of this sharing. And I want to emphasize something. I'm telling you about a program that could sound very academic. Every element of this program, every step of the way, everything is openly shared. Being an academic and knowing how things work, where I work on a study, I eventually publish it. In fact, when you read something in the news, that's actually old research. We've known about it for a while as academics. So we go to meetings. Well, we used to go to meetings, now we can't because of COVID. And we've seen the signs. We did not want Answer ALS to be that private. As we generate data, it's provided to any researcher, any academic, or any company. Because the fact of the matter is, as academics, we may make initial observations. It's companies that actually generate the drugs. So we want to share with them. Every single one of those tools that we generate, all of this data, all the genome data, we share with other researchers so they can use it. Even while we wait to figure out what we're going to learn from it, we want them to learn as well. Along the way, because we collect patients' biological fluids, such as plasma, some patients undergo spinal taps, all of that, but those biofluids are banked. It's the largest longitudinal biobank ever in ALS history. We've got more than 50,000, well, this slide says 60,000, so I'm a little out of date, biosamples saved from patients over that 1 to 2 year time period of following them. All of those biosamples, they're stored away for any researcher to use, not just us. Because as they begin to think about what pathways might be important, they might want to test them by looking at the biofluids that have been collected from patients. I mentioned earlier that we're now making those iPS or stem cells. We're well underway to making all 1,000. We're now well over 600. This is a very time-consuming part of the project, and this is the curve. And you can see we actually dropped off a little bit. Ed Rapp was unhappy with us along those lines. We do our best to stay exactly on that curve. Unfortunately, as many of you may know, COVID has been a major interruption to all medical research around the country. We will eventually pick up speed again as everyone comes back to the labs, which are happening now. But those stem cells, although we're using them for research, we give them out. And this slide shows that already 600 of those cells, already cell lines, that's 600 individual living biopsies from patients are available to researchers. And this map shows that we've shared them across the world already. So we're not waiting to figure everything out. We want to let others have a chance of figuring this out as well. And I'll probably mention this again, anyone who wants it. Now, again, this is any researcher. They have to agree that they're not going to try to figure out who the patient is. By the way, everything's coded, so you don't know who the individual person is. So there's no personal information revealed. Anyone can, any researcher or company can go to this website. They can search, I want a woman, 35 years old, who's had bulbar onset ALS. I want all the IPS lines. And they can order those lines from that website. And this is just a blow up of this website where you can really search. There are all kinds of search algorithms for picking the cells that you want and to be shared. And again, we have been sharing them. Okay, once we have those lines, now we have to start cranking through the analysis. We now take those lines and we analyze them for how does the environment affect the biology of your DNA? That's called epigenetics. How does the DNA convert to RNA, which is the nucleotide that follows DNA? We analyze all of the RNAs. And RNA is made into proteins. We analyze all of the proteins. In aggregate, with all of the clinical data, the app data, and all of that omics data, meaning proteome, RNA, whole genome, we generate about 6 billion data points per patient. That's a lot of data. And so the idea is to take all of that data and to begin to integrate it using artificial intelligence. We're doing it with my colleague Ernest Frankel at MIT. Again, and I'll show you in a moment, we're providing all of that data to others to analyze. But the idea is to take all of that data, crank it through the this cartoon gears, take an existing literature that we already know about ALS and begin to look for, are there actually subgroups of different patients that exist? Do some patients have a problem in proteins that aggregate? Do some patients have a problem with their nucleus, the cell, the core of the cell? because some patients have problems with things moving down the axons, the lung nerves. This allows us to begin to analyze that. This is just another slide that shows we analyze the different omics, we integrate that and look for subgroups of patients. And this is just a fraction of this team. As I mentioned, there's more than 100 people working together on this program to integrate. Once we begin to get clues, okay, so that's a clue, but it still came from this human cell. How do we begin to be sure it's true? So now you begin to try to validate that. Well, one way to validate, so let's just say we think a protein X is important in a subgroup of patients. I don't know what that protein is, but let's just say this analysis tells us that. Does that mean we're ready to go to trial? That's not really good science. I want to know now, do I really find that problem in patients' brains? So we have an autopsy bag, and we begin to see, do we see abnormal proteins in a subgroup of those patients? We can even take artificial animals, flies, and begin to say, does that gene affect flies? So we use model systems. We can go back to the iPS cells that we started with and said, okay, this patient, we think this pathway is abnormal. What if we tweak it? What if we turn off that, what we think is the bad part of the pathway? Do the cells look better? So a lot of ways of what we call validation. Knowing that, we're beginning to get ready to think about drugs and going back to our patients. One of the pathways we've come up with has to do with the nucleus. This is already emerging from the Answer ALS data. The nucleus is the core of the cell. And we've already begun to identify pathways from Answer ALS. Now, we by no means figured it all out. We're looking for those early hits. We actually already know that some of those hits are going to be the basis for the drugs being used in some of the platform trials. And already some projects are emanating from that big, vast amount of raw data. There are multiple projects that are being done by the Answer ALS Packard group, by other researchers who have taken our data, beginning to do their own research projects. So even though we're not done generating all 1,000 cell lines, there's enough data that people are beginning to take hold of it and using it to understand subgroups of ALS. So here's an example of one. Every cell has a nucleus. That's a little like tennis ball within a tennis ball. That's the computer of your cells. It controls everything, and it's got little holes in it called pores that allow things to move in and out. And we've learned in ALS, I may have lost the image. Yeah, we've learned in ALS that these pores in a subgroup of patients, they're missing pieces of it. That's actually shown here. Here's a normal patient. Those little green dots, those are those nucleus. This is the nucleus, and those are those pores. In ALS, they're gone. But because we've learned some of these pathways, we can give certain drugs that completely fix that. So here's one of those examples. You give the drug and all the pores come back again. That drug is now in clinical trial in a subgroup of ALS patients with the C9 mutation. So we're already beginning to jump on some of the data using these model systems to move into patient trials. In fact, there's already 2 clinical trials underway with Biogen based on that kind of early data from Answer ALS model systems. The C9 antisense and another drug just starting with Biogen that affects not just C9 patients, but it might be useful even in sporadic patients. Both of these trials are now enrolling. Ignore the Hopkins. This was originally an internal lecture. These are enrolling at multiple sites around the country, actually some of them around the world. So we've been also releasing this data. I've told you this, and we've given this data out to researchers around the world. We have a special portal where we've released the first over 100 complete data sets for all of those omics, the epigenome, the RNA, the whole genome, that researchers can begin to download. And they're starting to use it. Multiple researchers around the country are starting to use that data. So where we are today is that we've now fully enrolled all 1,000 patients. I'd be more than happy to enroll more, but it was our starting point. And quite frankly, I'll tell you in a moment, this was fully funded by philanthropy. It's not an inexpensive program. We've already sequenced over 800 of those 1,000 patients for their genome. We generated a program where we give their genetic results back to those patients. We've now made over 700 IPS lines, 600 before. As I move through the slides, things just keep moving. And those lines are already available for researchers. We've already begun to identify some subgroups. We have a lot more work to do. We have an app that's being used in patients. We have a portal, and with the help of Microsoft, we're really improving that portal, and we're publicly releasing that data. Here's an example of our first iteration of a portal. Some researchers can say, I want ALS, I want their spouses, I want men or women, I want ages, and I want to download all the protein data. I want to download all of the RNA data. All of that's doable now with a very simple agreement. There's no legal hassles for any of this. I really want the data fully available. And that new portal with the help of Microsoft is coming soon. And that help of Microsoft is really a wonderful collaboration with Team Gleason and Claire from Answer ALS, who has been helpful to make this move forward. So we're also, as I said, we're really working hard to make sure that researchers around the country have access to it. But that's not the only way. It's still sort of a limited information. Do they hear webcasts that we do? We're now working with the NIH and ALSA to find ways so those organizations can make opportunities available to use our data for free, if you will. So we're asking NIH to do what's called an RFA, request for applications around Answer ALS data. They're big research grants, but they're grants based on using our data. We're working with ALSA to do the same thing. We are closely working with the Chan Zuckerberg Initiative. I'm actually funded to do some of the work with them. And we're also talking to other dementia groups, because ALS is not just ALS, it overlaps with dementia, hoping we can partner as much as we can to move our data and help other diseases as well. Now, the last few minutes, I just want to tell you about some exciting new things that many of you have heard. The concept of subgroups has now been really validated with 2 publications that came out just this week, focusing on SOD1. Now, here's an example of a timeline. It took a long time. The SOD1 antisense trials, turning off the gene for SOD1. We learned about SOD1 in 1992. It's now 2000 that we first have our first set of trials. As an example of how things move faster, in 2011, we first learned about C9. That C9 trial is already underway. So we've at least half the timeline, probably more like a third of the timeline. That is, we're that much faster in carrying out trials, because when we know the subgroups, we can very quickly develop drugs that are targeting genes. And this is a great example of why targeting therapies are important. ASOs as a particular candidate are very important because they're very good for orphan diseases, most very rare diseases. For example, it could be the subgroups of ALS patients. There aren't many of them. Maybe a biotech company won't be interested, but this orphan disease approach with ASOs are a very powerful approach, and the FDA is very keen on these small number of patient trials for these rare subsets. The downside, this is not going to help most of my patients with sporadic ALS, but it's going to help some as we learn about whether they're big common subgroups or small subgroups. This has also been done in a rare form of ALS, FUS ALS, with a group at Columbia, also treated a single patient with a different kind of gene therapy. So this advent of really understanding subgroups and having very targeted therapies, we're just on the cusp of this. And this will expand. It's being now expanded to other rare diseases as well. To do this, we are really developing these gene therapy clinics. This is happening in kids, and probably the more we sequence children, we'll begin to individualize therapy. For all of you, and for all of you who even don't have ALS, the concept of whole gene sequencing in all patients will be something in the future. So every patient will know their gene sequence, will know their variants. And somewhere in that grand plan, there has to be a national database, the way of commercial databasing. So you get it's like you get your MRI and it's stored somewhere, you get your gene database, and every couple of years they look back and say, well, there wasn't a gene identified in 2020, but it's 2025. Now we know one of your genes are abnormal, and we've developed a therapy for it. This is the future of this kind of subgrouping of medicine. And we certainly think this is approachable to ALS as well. I'm going to skip through this slide. My last slide is this. This is why it's so exciting in ALS. This is actually a spreadsheet. I don't expect any of you to really read this. This is just Johns Hopkins. We have 25 trials coming up in the next year. This is massive. I've been doing trials, as I told you, since 1989. I've never seen anything like this. We have a platform trial. We've got multiple biotech companies. We've got multiple big pharma companies in this list. There are many opportunities for patients. Some of these are, if you will, the tiny trials, just C9, just SOD1. Some are for many patients. Big targets today are things like inflammation as one of the big targets. And we can talk more about that. But this is exciting. There's so many opportunities for patients. And buried in this chart, and you can't read it all quickly, it's not just that we're trials. Some of these, it's a 1-to-3 ratio. It's 1-to-4 ratio. It's not the old 2-to-1 or 1-to-1 ratio of placebo to drug. And most of these have open label phase. Some of them even have expanded access. This reflects you as a community pressing and we as physicians pressing on companies. I do not like companies that come to me with a 1-to-1 trial. I don't like companies that come to me with no plans for open label access. In fact, most of us won't do trials unless we're desperate for that company that don't provide some expanded access or at minimum open label. We really demand this for patients, and as you are demanding, and rightfully so. So I want to end. I took up way too much time, but I'm happy to stay on as long. I've been talking, but this really reflects just a huge consortium of wonderful collaborators, clinical, basic science collaborators around especially the Answer ALS program. And the other part of the slide shows the many groups that have come together to help fund Answer ALS. Answer ALS is not really government funding. It's so pie in the sky, the government would never fund it. And that's not, I'm not throwing spears at our NIH. I sit on those review panels. It's a different kind of biology. But to get to the point where we really have these novel hypotheses, you need philanthropy to jumpstart this. So Caterpillar, American Airlines, IBM Watson, Microsoft, Team Gleason, and on, Jen Zuckerberg, F Prime. Many groups have really contributed to make this kind of program possible. And what will follow will be the research around these hits. Then we'll go to the NIH. Then we'll go to other groups. And hopefully pharma will step in and begin to move some of this into drugs. I'm going to stop and take questions.
Thank you very much, Jeff. That was excellent. And that was a lot of material too. And I think one of the things you've always focused on, and that's been your mantra, is collaboration. And it shows that, how you've been able to bring in a lot of research, a lot of external partners into what you're working on to really make this happen. So with that, I think we can go into, we had a lot of questions in our chat, and I will introduce Tara Tadimala, who is here. She actually graduated from Case Western pre-med and getting into medical school, and she's also my niece. So have personal experience with her uncle having gone through ALS, and also Sarah Diaz, who is a medical student at Washington University, and also, unfortunately, she had, her dear father was affected by ALS, and she's here today going back to med school to really find the answers for ALS, and very dedicated. So I'll introduce both of them, and Sarah and Tara, you please take away with the Q&A.
Awesome. Thank you, Dr. Rothstein, for speaking with us today. Once again, my name is Tara Tadimala, and I will delve right into the questions that we had. So the first one, what is your opinion on studies that show a deficiency in resistance to growth hormone in ALS patients?
So I don't have a strong opinion. Growth hormone had been studied years ago in ALS, and many years ago, before my time even, there have been studies using various kinds of growth hormones. I guess I would have to say, so again, remember I told you at the start, I'm blunt. I don't think there's really good sufficient evidence to support that as an approach to treating patients. I'm not going to tell you your physician shouldn't do it. I'm not going to tell you it's malpractice. I just don't think there's enough science around that to be convinced that it's worth an approach. But I'm going to tell you I don't think there's enough science that teaches me that that is a deficiency in patients. Keep in mind, for all I know, there could be a subgroup of patients that it's a major role in. We often start with population studies to understand everyone, but there's no reason to believe that there couldn't be subgroups of patients that have that as a problem. I just don't know of any strong data that supports that.
Thank you.
Hi, Dr. Rothstein. I'm Sarah. The next question kind of goes along that. So based off that answer, what, in your opinion, in clinical trials and new treatments do you see most promising and potentially bringing on a treatment or a cure in the near future? You knew you were going to get asked it, so. That's
okay. My clinics are Monday morning, so believe me, I get this every Monday. So I showed you a long list of trials. Now, most of those trials fit in several categories. There's a huge interest. I'm going to tell you the pluses and minuses of what's exciting. Right now, there's a huge interest in inflammation of the brain. It's called neuroinflammation. It's not autoimmunity, to be clear. It's inflammation. There are cells in the brain. And every disease gets this. MS, ALS, Alzheimer's, Parkinson's, Huntington's disease. They all get inflammation in the brain. As the neurons begin to die or something goes wrong with them, the glial cells, astrocytes, oligodendroglia, spark, if you will, being a little hand waving here, microglia, which are the endogenous immune cells of the brain, to get activated. And some people believe they're propagating the disease. They're sort of pushing it forward. And to that extent, or whatever, there are numerous drugs. There's C3 complement drugs. There's C5 complement drugs. There's CD40 ligand that are all being targeted to those pathways. I have no idea which drug is better. Now, one of those, the CD40 ligand, years ago, studied by Biogen, in the early version of that drug, was killing people. Not good. The newer versions presumably are safer. Don't know. That's why we do initial trials. That's a huge push right now, is to minimize inflammation. Now, I'm going to add one other very important point. I'll do it quickly. The earliest trials in ALS back in the early 90s that I mentioned a lot of drug trials. And actually many of those drug trials were anti-inflammatory. Intravenous gamma globulin, methotrexate, total lymphoid irradiation, steroids. All of these were drugs that we as clinicians very commonly use for autoimmune diseases. They're really easy. We all know how to use them for diseases of muscle, blood vessels. And so because we had them, we said, ALS might be autoimmune. We threw them at ALS. These are trials from about 1989, 1988 to about 1995. Okay, they failed. Obviously, we'd be using them. So earlier iterations of anti-inflammatories had no effect on the disease. These are more sophisticated drugs, so maybe they'll be better, or maybe they won't. I'm willing to definitely, so we're running multiple trials like that at Hopkins, and I'm definitely willing to try them because other drugs aren't working. These are sophisticated versions. Most of these are being run, not all, by big companies that know what they're doing. These can be very toxic drugs. I mean, really toxic. Masitinib is another one. Some of these drugs can cause horrible infections. So we're going to have to watch patients closely, but they are the wave. Then there's a couple other bins. There are drugs that are being developed that affect protein aggregation. There aren't a lot of them right now in trial. There's one by Clen that's going to be part of the platform trial. It's about metabolism. We've known about metabolism errors in ALS. It's a wacky drug, but who knows? Sorry, I throw out occasional adjectives here and there, but I'm willing to try these drugs. So most of the trials I'm seeing run by, say, the platform group, which are already being looked over by academics to sort of say, did they have reasonable research? Most of those don't have a lot of research. Most big pharma always will have a lot of, big pharma typically have a large package of research that says, we're willing to spend that $1 billion for that trial. Little pharma often doesn't have much research. They do one mouse study, a fly study, a fish study, that's about it. And in the old days, we weren't picky. We'd say, shit, my God, I'll take anything. I need something for my patients. Today, we can be a little more picky, in my mind. So I hope I answered your question.
Thank you.
For the next question, could some of the drugs be tested for familial ALS potentially benefit patients with sporadic ALS?
Yes. It just depends upon the pathway. So the drug that we just studied, New England Journal paper by my colleague Tim Miller, that's very specific for SOD1 mutations. There's not a lot of strong evidence that that drug would have any use in sporadic patients. There's a little bit of literature that might suggest that, not strong literature. There are drugs being tried in C9 that affect the nucleus. One of them is very specific to C9. One of them is so non-specific that it's also being used in sporadic ALS. There's another drug online for that's also kind of very targeted, a little more maybe for molecular forms of ALS called Ataxin-2. It's an upcoming drug trial from Biogen, but it will be used in sporadic as well. So there's no question that, but it's going to be on a case-by-case basis depending on the particular drug. And all of you should, there's lots of experts out there. When you have ideas, you should definitely talk to your local neurologist or maybe scientists in the community who might help educate you. By the way, I'm happy to answer questions by email.
Thank you.
But one last thing about email, if you write me, I type like a third grader. I really have lots of spelling errors because I hit send right away. So I apologize in advance.
Good to know. In terms of the Answer ALS research, and has there been any look into the differences in the neurons, either the degeneration or process that they go through between slow moving or slow progressing versus fast progressing ALS?
Great, it's a great question. We've not done that. So the iPS cells, the system I described to you, no one's done that yet. We're mostly in the data generation phase. We call that phase 1. Phase 2 is when you start saying, I've got the data, now I begin to test hypotheses. That phase 2 will be run largely through the Packard Center or anyone else that gets the data. And we'll begin to essentially fund research grants. That's a great question. So someone takes 5 lines of a really slowly progressive patient, 5 lines of a really fast progressive. What are some of the phenotypic differences there? So you're absolutely right. Great question.
Awesome, thank you. For the next one, could the new government funding and easing of FDA regulations help with the research to find new remedies or cures and approve them a lot quicker?
So I can't tell you anything will lead to faster improvement in faster approvals. Funding always makes a difference. More funds brings more researchers in. So it always makes a difference to speed things up. There's a long history in almost all of medicine. Throw money at a problem, you get more researchers with different ideas, you'll get answers quicker. That doesn't necessarily speed up the pipeline because what I discover in the laboratory still has to be converted to a drug. I'm not a drug company. And so somewhere in that equation, you've got to get companies interested, biotech, pharma, who eventually turn that academic discovery. Because in today's world, not too many companies do their original research. They rely on our findings and build on them as they develop a drug. So that's important. The speed by which the FDA moves, it's not really that slow, quite frankly. The requirements are there. You got to know that a drug doesn't kill every other person, every other animal before you give it to people. It's just a requirement of all therapies. And one of the reasons is, and I don't want to get to the politics of all of this, because remember, when a drug is approved by the FDA, any physician can use it for anything they want. Okay, so if a drug's approved for ALS and you've got, I don't know, toe fungus, your physician can use it for your toe fungus. It doesn't matter, it's FDA approved. You might be wrong to use it for that, he or she can do that. So if a drug turns out to be really toxic, they give it to you for your toe problem and you drop dead, yeah, not good. So they need to know about safety profiles of drugs. It's long been a requirement. So I'm not going to get as much into that. So anyway, but things are quite, I mean, from when I started, things were moving a lot faster. By the way, one other point, when I came on, you guys were talking about expanded access. Something you should know, it's great that there may be funds for companies that want to provide expanded access, but there's part of that equation many of you don't know about that I've been dealing with this from some of my patients. Because expanded access doesn't mean suddenly the company can give it to all of you. It still has to go through typically an academic, which means if I'm going to give you an unapproved drug yet, I have to first get an IND. The FDA, I've done the newer paperwork. It's fast. I still have to do it. The company has to give me all of their literature. I hand it in for my single patient to the FDA. Doesn't take too long. Then that's approved from an IND. I now have to go to my institutional review board because it's still an experiment. And I have to get FDA, IRB approval. That takes time. IRB doesn't like the way I wrote it or something, or something about what was given, comes back to me. I give them back something, comes back to me. And everyone who does clinical trials knows that. So there's a part of that equation. There's no way around that. That regulatory element is not going away. So expanded access is great, but there's still an administrative part, puts a timeline on that. And it all depends upon the company giving me data, the FDA responding, and my IRB. And my IRB at Hopkins might be fast, and Mass General might be slow as all molasses. You don't know that in advance. So you just should know that. There's other elements to that. It's not just the company. And of course, most importantly, the company has to be willing to do that. Most companies aren't willing to give expanded access. Just because it's available doesn't mean it actually happens. The company has to be willing to do it. So, other facts.
Thank you.
Questions?
So in terms of that expanded access, is there something either patients can do? Is there something that our population can do to maybe help with this in terms of rare diseases and ALS in general to make it more approachable with the big pharma companies?
Yeah, so great question. So I don't know. So pharma is its own bin. You can pressure the pharma. It's an internal business decision on their part. The money helps because pharma sometimes will say, yeah, I don't want to do that. It's going to cost me extra money. I don't have that money. There are different reasons, and I'm not going to tell you I know all the reasons. And I just told you the administrative reasons. So in a discussion with IMALS recently, there's a board of us that are advisors. I brought up the idea, well, what if we had a central expanded access group that does the work for me? I'm going to, because we're all busy. We're running trials, we're running laboratory research, and then suddenly I've got to do an expanded access. That's additional. And by the way, just to add, again, an honest part, when I do a trial, the companies pay us for all that time. By the way, it's not like I make money to buy a car or something with it. It's just they have to pay for time. That's their responsibility. Expanded access, no one pays for that. Now, you should think it's the goodwill of your physician, but it gets to be a lot of time. So one idea I discussed actually with IMALS was, what if we actually had a central group that was willing to do that for the physicians, for the clinicians? They do a lot of the paperwork for them, or they help out. And it would unburden us, and it would actually then get things faster, because maybe I'll get an email from the IRB, oh, would you fill this out? Yeah, but I got 3 other things. I've got to respond to my patients. I got a clinical trial I've got to get going, and it gets left behind. What if I could actually turn to someone and say, look, I just got this query. Can you deal with that and fill it out? And then I'll send it back to my IRB. This is what I do with my staff all the time when I do trials. So the idea of, if you really want to speed things up, why not have a central group or a couple of people around the country who act as that expanded access facilitators? So that was just an idea we thought of.
Yeah, no, that's wonderful. Thank you.
Thank you. For the next one, what is your opinion on stem cell treatments for ALS? Do you think NurOwn is showing promising results, and can it help some people?
So you're asking about results when trials are not done. So I've told you I'm blunt. Until a trial is really proven, I don't know if it works or not. Remember, I just told you the history. I've been doing this since 1989. I've seen trial after trial after trial where someone says, oh, this is fantastic. Oh my God, this is great. I'm on the drug. Actually, it turns out you're on placebo, because we learn afterwards. Oh, this is the best drug ever, 1995, 2005. I have no way of knowing. Until a trial is done and fully vetted, I honestly don't know. If you told me I hear about stem cells and I want to go to Israel, and if I pay them $500,000 to let me in the trial, run as fast as you can away from that. I've had trial patients do that for trials in Ecuador. $100,000, go to Ecuador and get my immune system replaced. Don't ever do stuff like that. That's a mistake.
Okay.
Other questions?
In terms of trial models, and just back onto the Answer ALS model of collecting the data but then being willing to share it with other researchers, is there any other organization that's running through that system where they're collecting large amounts of data but then saying it's free and accessible to anybody who would like to use it in their research?
Yeah, it's a good question. Okay, so historically in ALS, the only other version of that comes to mind quickly was when, oh God, I'm blocking on the name. It was PROACT. It was a collection of past clinical trials. It was put together. Melanie Leitner ran it. I'm blocking the name of the organization that sort of doesn't exist anymore. Maybe someone will remember what it was, but they collected clinical trial data and made it available. So it's a very valuable set of data, past clinical trials. So it's not biological data for the most part, it's clinical data. It's been used to help model future clinical trials and design. I've never seen large amounts of biological data, small amounts, but large amounts of biological, I haven't seen that yet. Now, just to be clear, this is starting to be done. Actually, the Answer ALS is now being modeled by other disease organizations. So in Parkinson's disease, there's called AMP-PD. They're not doing iPS cells, but they're doing blood biological information. And that's being done in part through the NIH. There's AMP-AD. Actually, I don't know what AMP stands for. I should. But AD is Alzheimer's disease. That's being done again through the NIH in part. And that's for biology around Alzheimer's brain samples. No one has done it on large scale around the equivalent of a biopsy, iPS cells. Partly because it's, to be fair, this is so far a $42 million project. It's a lot of money. And again, thank God for all the people willing to donate towards it. And another reason why you're going to put all that money into something, darn it, you better well make sure everyone gets that data. It shouldn't be held by one person.
Okay, thank you.
Awesome, thank you so much. The next one, is riluzole as effective for lower motor neuron disorders as on upper neurons?
Say that again, sorry.
Is riluzole as effective for lower motor neuron disorders as on upper neurons?
Yeah, so we don't know much. It's a really good question. And clinically, so just so you know, clinically, it's very easy to detect and measure and quantify lower motor neuron. Those of you who don't know what that means, those are the motor neurons in your spinal cord, which when they're diseased leads to muscles that are atrophied and twitching and very weak. Upper motor neurons gives the stiffness in muscles and the brisk reflexes. There's not good quantitative tools. And so there's really never been a trial specifically focusing on upper motor neurons. And again, to be fair, ALS involves both neurons, but upper motor neurons do not cause death, the dysfunction. It's lower motor neurons, because the lower motor neurons drive your intercostal muscles, the muscles between the ribs, your diaphragm, and what are called your strap muscles. Those three groups of muscles are necessary for breathing. It's lower motor neuron derived. And when those don't work, you don't breathe. And so a great focus always has been on lower motor neuron more than upper motor neuron. Thanks, Tara.
Thank you.
In terms of C9 mutations known to be like higher populations in European, in general, the European population, are there any other studies that are looking at population, ethnicity population in terms of familial or sporadic genes?
It's a good question. So I have to tell you, that's not my expertise. There may be, and it's just, it's a blank zone in my brain. I can't answer that. Someone like Brian Traynor, who's a geneticist at NIH, would be a great person to answer that question. I don't doubt there certainly have been over the years, but currently I don't know.
Okay, thank you.
For the next one, what are your thoughts on Capri ATSM as a potential treatment for ALS?
I think it's a great idea for SOD1 ALS. So NEALS, many of you may know who NEALS, the Northeast ALS group, we have a science advisory committee. So I run it, Bob Brown, and I co-chaired it more recently, Tim Miller. And what we do, now there's a whole group of us actually, it's more than just the three of us. We evaluate, companies come to us and show us their drug candidates, and we try to tell them, how do you get the best information to run a clinical trial? What do you need? What's missing? And then we also help them design trials. We move from the preclinical to the clinical. The reason I'm telling you is that that group actually came to us for advice about clinical trial. We were very clear to them. It's a great drug for SOD1 ALS. There's absolutely no good reason to study in sporadic ALS. They didn't necessarily take our advice. Maybe they're right, but so it looks to be a drug relevant to only SOD1 ALS because it helps. It's around the biology of SOD1, which is mutated in only a small number of patients.
Thank you.
In terms of the ALS Association in other countries, are they potentially able to be part of studies that are going on, either the platform trial or other studies? We have a couple of international patients on our call interested in how to gain access to some of these trials and treatments.
Yeah, so okay, so it all varies. Platform right now, and Merit, of course, Merit Skovic could answer this better than I. I believe right now it's US based. Whether or not there's a Canadian site or two, it could be, but you're really talking about Europe. Most other trials, when they're run by pharma, it's up to the pharma company. So currently on that list I very quickly threw up, many of the trials are actually in Europe and US. Now, when I say in Europe, that doesn't mean every country. You live in Spain, you might be out of luck. You live in the UK, there are typically sites. The most common sites in Europe are going to be UK, France, and Germany. By the way, this is off the top of my head. Often they're not in Japan because the regulatory environment in Japan and running trials are much more complex in Japan. So more often than not, they'll be, when they're international, be US and United States. I don't even think they're commonly done in Australia at the same time. Sorry for those of you who might be from Australia. That doesn't mean there aren't trials, but many of the big pharma run the trials. So platform for right now is US. Now Answer was initially completely US based, and then we opened up the opportunity to bring in sites from Canada. But as I told you before, Answer was kind of expensive, so they had to decide if they're going to pay their own way, and they had decided in the end, if I'm correct, that they're going to do their own smaller version of Answer within Canada. And I don't know more about it than that. And we've had discussions in Australia. They've on their own decided to do their own Answer-like program that they've been starting up with their own internal funding. So we share our model with everyone. Why not? And I don't think there's an equivalent in Europe.
Awesome. Thank you so much. For the next question, can you reverse ALS and redevelop neurons that are damaged for those who cannot swallow or eat or talk? More generally, can you grow nerves back?
Yeah. Okay. The blunt answer is no. The nervous system cannot regenerate. When a motor neuron dies, it's gone for good. Now, if it's slightly damaged, it probably can come back. So I'll get to reversal in just a second. And a great example of that is in the second paper in New England Journal. My colleague Bob Brown used viral vector therapy. I've seen the videos. And then one of them was a young man, if I remember correctly, who no longer could move his foot. And with the therapy, regained some of that movement. Now, what that probably meant was that in the time frame of the experiment, because just so you know, motor nerves grow very slow. You've got to remember, it's like 1 millimeter a week or something. It basically, if you had to regrow a motor neuron from your spinal cord to your foot, it would take 3 years to regrow. That's just the human nervous system. It takes a long time to regrow nerves. But if a nerve is there, meaning the cell is there, and it stopped firing, so let's face it, if we all get sick and we're in bed, we're not getting out of bed, that means our neighbor doesn't know we exist anymore because you're stuck in bed and you feel like poop. But you get healthy, you're outside again. And motor nerves are like, motor neurons, we think are like that too. If the motor neuron's injured and it's not firing, it means no contraction. But if it gets healthy again, you regain contraction. The reason I said that, because in Dr. Brown's study, he actually saw the patient, although the patient eventually could not recuperate, regain some motor movement. And that was our first proof in humans, forget about mice, that actually there is some recuperability, which means, if I really extend that, that means if I had the best drug today, take away cure, I don't know how to cure ALS, but if I could stop it completely, some of the motor neurons are actually sick, and if they regained function, patients would regain some function as well. Again, that's an honest answer. So when patients say, can I get back to normal, where I was before I ever had illness? Right now, there is no science that allows that. That doesn't mean someday there won't be. In the early days of stem cells, we used to think that was possible. I don't think that today. But that doesn't mean I can predict what's going to happen 5 years from now. None of us can. But the newest science from that SOD1 trial says, I bet people could get better somewhat. Now, is that a full reversal? No. Have I had reversals? Because I know Dr. Bedlack has talked to you. I've seen probably over 10,000 patients in my career so far. It's a guesstimate, at least 10,000. I've had 2 patients who've gotten significantly better. So 2 out of 10,000. I have no idea why I got better. They've also been lost to follow-up because they don't need to come back to see me anymore. Now, did they get better because they got better, their ALS got better, or did they have something else? It looked a lot like ALS, because 2 out of 10,000, that's really uncommon. So when my patients, I'm telling you the same thing I talk to my patients about. The honest answer is I don't know how to reverse ALS. And no one does, there's no drug. What I care about is finding drugs that really stop the disease and then allow that natural partial reversal. That's what I'm looking for. I am looking for the cure, but I have only a certain number of years left in my life, so I'm being practical.
Great, thank you.
You mentioned during your talk the 391 whole genome sequence data. Is that going to be released to some of the patients too?
Yeah, so I went through that quickly. And again, I'm already way over time for many of you. I apologize. What we did, when we started the study, a little nuance to a study. When you do a research study, it's research only. You're not allowed actually to give, by IRB rules, the data back to patients. It's for research. And again, there's nuances in that. When we did whole genome sequencing, so when you get a blood test done, it's being done in a verified clinic. And every clinic that does a blood test, maybe it's your blood glucose or something like that, that lab has what's called CLIA certification. There's a specialized society that says, you know what? Every time that test is done, we verify that it's an accurate test. When we did whole genome sequencing, it was not done under, quote, CLIA certification, which meant no matter what we find, we're not actually allowed to give that data back to patients. It's not CLIA certified. On the other hand, if we find something abnormal, we could just simply retest under a CLIA certified retest. That's exactly what we did. When we found abnormalities and when patients gave us permission, they said, oh, I want my data back, we would retest. And instead of 8 centers and hiring 8 geneticists to talk to patients, we did something very novel. We hired a central geneticist, Jennifer Rogenbach at Ohio State, and she became the focal geneticist for the entire nationwide program. She would contact patients. And by the way, I know there's been other genetic studies by other groups, not academic groups, by other companies that say they'll give data back. If you're going to get genetic data back, God damn it, you better have a certified geneticist who knows how to talk to you about what those genes mean, not some student.
Absolutely.
And so have geneticists, sorry, not students, do this. And so now we're giving that data back.
Okay. All right. In terms of, you did mention time, so we just want to make sure that we are respectful of yours. Do you have a limit? We have, looks like about
tonight. You're my.
Okay. We looks like we got about 10-ish more questions. So as long as it's okay with you, then we'll keep going.
Great. Our next question is, if we focus on personalized medicine, how do you feel about a p-value of 0.05 and whether we should adjust sample sizes for clinical trials?
So P05 is the universal standard for the first phase, if that's what you're asking. Everyone accepts P05 as the acceptable, meaning that means there's a chance that 5 people out of 100 you're wrong on versus 95 out of 100 you're correct on that assumption. That's pretty good. That's universal, both preclinical and clinical. Anything less than that, then people sort of worry that maybe it's interesting or maybe it's just fake. So that's standard.
Great, thank you.
There's some studies out there that are showing complement is dysregulated in ALS. What is your opinion on C4 of complement in ALS?
So there's no question, there's lots of studies in ALS that show that the immune system is kicked up. C4 is one of those pathways. This is actually why, that's some of the human preclinical, and that's important, I said human preclinical. Whatever I do in mice, and I consider iPS cells better than mice, but they're still not you, the patient. When there's human data, and by the way, not human data in 3 people, but human data in a large number of people, that's important. That means something's going on. Now, the kicker is that an event? So one, it tells us there's an immune activation. The immune system is being charged up by degeneration of nerves, by muscle, by the brain. What we don't know is, and I probably should have showed the slide. If I were to show you a slide, and I won't pull it up now, in our studies of both patient material and preclinical work, iPS cells or animal studies, we know that there's at least 10 different pathways: mitochondria, nuclear biology, glial cells, transport down the axon. All of these pathways go wrong in ALS. What we don't know is when I see a patient, were all 10 of those pathways activated? Or 3 of them activated? Were they activated when they were 20, but now that they're 50, they're no longer activated? So the temporal sequence of when that pathway is activated and which one of those 10 are active, I don't know. What I do know is very end of the disease, inflammation is activated. Now, what I don't know is if I fix that inflammation, is that one of those 10 pathways? Will it make a difference anymore? Because 9 other pathways are all screwed up in the patient, and inflammation is only accounting for 10% of the injury to the patient. We don't know that yet. We have no way of quantifying the degree of injury. So I like car analogies. Some of my patients know this. You have a car, the carburetor's not working, it's got a lot of rust spots, the fuel pump's not working. Well, some of those count, they're all defects. Some of them don't count at all. A little rust on the car, a scratch, dent, who cares? It's a defect. Fuel pump's not working, car's not going anywhere. And so in ALS, we don't have a way of weighting the degree of injury. So inflammation, we can really see how important is it to the course of the disease? We don't know until we try those drugs. And so that's the important part of trials. That's why we want to study them, but no one really knows which one's more important.
Great, thank you so much. On the next one, there are many people who have concussions, and anecdotally, those PALs seem to have bulbar onset at a much higher ratio. We know that NFL players with concussions have 8.5 times increased incidence of ALS. What do you hypothesize we are missing in the rehab of concussions, and how do we address the pathophysiology before the onset of ALS?
Yeah, so there's no question there is data on trauma, and trauma induces may induce certain pathways in the brain. There's been a great focus on something known as tau, a protein that's not commonly found in ALS, but it's very commonly found in trauma to the brain. And I think I don't actually know that answer. I don't, period. It's a good area open for investigation. And beyond that, I think we're left with those, we'll call them epidemiologic studies, but no good mechanistic studies. And that needs to be done as well.
Awesome.
By the way, very early studies of trauma in SOD1 mice, I'm remembering, I've got this long history of seeing all this research, not my research, by the way, and trauma actually didn't do much to the mouse. It actually been studied. at least some early mouse studies. So if you actually just cut the whole nerve of the mouse, the motor nerve, you'd think, wow, does that make him, does that slow the disease or to make it go faster? It didn't do anything. That's not the same as trauma to the brain. And there've been a few studies, but not a lot.
In terms of Radicava, can you just speak on your thoughts on that?
FDA approved drug, a real pain to use. I mean, everyone knows this. We were okay. Let's go back a step. We were really excited we had a second drug. Really excited. We had Riluzole for 2 decades. Okay, it's a bit of a poopy drug, not very strong, but it's there. Reliable, meaning reliable clinical efficacy, not something my patients can see. I'm very clear that I educate them. I'm not going to feel better on it. You're not going to feel better on Radicava. Keep in mind, the Radicava trial was largely on top of patients who already were taking Riluzole. So when I see patients, I don't want them to take Radicava without taking Riluzole. I think, I don't know the exact numbers, but of the 66 patients, 60 were on Riluzole as well. You really need both drugs to have any hopes of efficacy. The problem is it's an IV drug with a weird therapeutic regimen that none of us really fully understand, quite honestly. You do it because that's what FDA approved, and that's how they had studied it, 10 and 14, these crazy dosing schedules. The only thing good about all of that is one of the trials on my list that many people are going to take part is they're moving to an oral form. Thank God, a pill is far better than IV therapy. And it's actually a simple trial. And I have 2 versions of that. One, I'd love to see it replicated in the United States. Does it really work in the US? It was studied only in Japan. I don't know if that's the same as the United States. All the company has to do by FDA rules is just prove that the drug levels of the oral form match the drug levels of the IV form. So it's a simple trial, not a long trial. But if it works, it's nice to have a second drug. Obviously, we want much more powerful drugs, but there was enough evidence to say it worked, and therefore I support it. It's just, and by the way, the other thing I didn't mention, and all the physicians I know balk at, it's ridiculously expensive. I mean, ridiculously expensive. There's no excuse to charge that much money for a drug that's been studied in Japan since like 2000. They've been around. Companies will often tell you, I got to charge that much because I have to recoup my costs. For that drug, complete nonsense. I don't recoup any costs. So I'm sorry. But there's no question, when patients want it, we will give it. It's my job as a physician to make sure patients have access to drugs and therapies that are proven, whether I like how well they work or not. When Riluzole first came out, a lot of people wouldn't give it. They said, well, I saw the trial, no one got up out of the wheelchairs. That's an unrealistic view. It's a hopeful view, but it's an unrealistic view of what a drug can do. And so I think we have to respect the science.
Absolutely. Just to follow up on that, because we do have some patients that are newer to the ALS population, and we did have a follow-up question that said, what is the difference between Radicava and Riluzole, or just a general basis for someone who doesn't understand or doesn't know yet?
So Riluzole works at one of those, remember I told you there are like 10 different pathways? One of the first ones discovered was what's called glutamate toxicity. Glutamate is a neurotransmitter. You're able to watch me because glutamate works in your retina. And it was found to be abnormally regulated in ALS, in sporadic ALS. And that led to theories that anti-glutamates, and Riluzole is one of the first anti-glutamates, that it blocks some of the toxicity of glutamate, not very powerfully, and it works. Radicava, it's not entirely clear. When Radicava first came out, I was actually at another group called Target ALS, and a rep from the company came out, and he wouldn't tell us what he thought the mechanism was. The literature suggests it's somehow some kind of antioxidant. But to be clear, many antioxidants have been studied in ALS without efficacy. So I'm not sure we entirely know how it works. But that was the presumption, that it's some kind of antioxidant.
Mm-hmm.
Thank you. Conditions like cystic fibrosis and cancer are treatable these days. In your opinion, do you think too much time has been spent working on finding cures while good therapies have been ignored for treatment?
I think, so I'm wondering, so cures and treatment in my mind are kind of the same. One, it's a therapy. A cure is the maximal therapy. A treatment may be marginal, like Riluzole. But then there's I'm not sure if you're referring to symptomatic treatments. So in Parkinson's, if you have a tremor, we have drugs that get rid of the tremor. They don't stop the progression of Parkinson's. And so they're symptomatic management. In ALS, we have a fantastic drug for the hyperemotionality that comes with bulbar onset. Bulbar onset patients sometimes have a disconnect in the brain circuit, and they very easily laugh or cry. They're not depressed. It's not depression. Years ago, we used to give antidepressants, wrong drug for that. But we know that circuitry enough that a combination of two drugs, when given together, and the combinatorial drug is known as Nuedexta, works very well at suppressing that symptom. Doesn't change, by all accounts yet, the progression of the disease, but it's symptomatic measurement. And that's the one drug. We don't really have good drugs for muscle spasms. We don't really have good drugs for muscle twitches. And so anyway, and that's what, I'm not sure if that's what they were getting at. That's my answer.
Thank you.
Do you know of any trials that are working with epidural steroids as a treatment for ALS?
I do not.
Okay.
No, I don't. Now, steroids, systematic steroids, meaning peripheral administration, were studied years ago of no value whatsoever. Very high dose, nothing.
Okay, thank you.
Yep.
BMAA is hypothesized to replace L-serine and SOD protein. Would copper ATSM be viable for populations of people who had BMAA exposure?
You know, it could be. I don't know who that population is other than in Guam. The BMAA story, or people who, there's a whole story about certain blue-green algae that may cause these kinds of toxins. I don't know if it's widely accepted. There's research around that. Beyond that, I can't add more to that argument. It's not there's not a lot of science I can hang my hat on that.
Thank you.
How long after top-line results would the timeline be for the access to patients, assuming top-line results indicate it is helping?
Ooh, so I don't know that answer. Because we don't determine that. If I'm extrapolating, I think you mean, you know, for trials done, a big trial, I guess, how soon do we find out? Well, when a trial is run by a pharma company, they're responsible for analyzing the data. They often convert, and at the time they tell us, so just to keep in mind, big pharma runs trials, but they don't often run it without us. In other diseases, like Alzheimer's disease, they'll run trials at private physicians. ALS trials are always run in academic centers that are experiencing ALS. And because of that, we're part of that consortium. When a company has now analyzed all of its data, let's just say Biogen or something like that, they then will have a meeting with all of us. And although it's supposed to be private, around that same time, they'll have to make a public release because they're a publicly traded company. And there's all these rules about when they find out something. I am no expert on any of that. So typically, it's once they've done all of those analytics that they tell us, and we have no control over that. And I don't mean they hide anything. It's their data. I mean, remember it's one of those funny things. You act as volunteers for trial. We act as we carry it out. Maybe a bad choice of the word is we're slaves to that trial, but we don't own any of it. That company paid for the trial. They paid for the drug. They own that data and they control it. We can ask them to do things. We can be polite about it. We can be really aggressive about it, but it's still their data, their property. And you actually agree to that when you sign a consent to be part of a trial. And we agree to that when we agree to run the trial, but it's really their data. And this has been controversial over the years because sometimes people, not for ALS, will say, well, the company suppressed that negative data. They wouldn't let it out, which is wrong. Many of us all believe that when a trial is run, regardless of the outcome, we should know the data. We've pressed companies for years. We've only got a little headway. When they're done with the trial, we want all the data because sometimes companies don't give you back the data. They tell you the calculated result. They don't give you all the raw data. We want the raw data so we can use it. And slowly companies are coming around to that. It's another thing you might want to pressure. You don't get immediate results from that, but in the long run, having all of that data is valuable.
Great, thank you so much. And our last question, what are possible explanations for the variance in onsets, bulbar, lumbar, FTD?
Yeah, really important question. One, I don't know, period. Even the mutant forms of ALS still can be, so C9 was originally thought to be mostly bulbar. Not really. Maybe slightly more common in bulbar. And so are those motor neurons in the base of the brain slightly more sensitive because of other gene variants? Other biologies about those neurons versus the neurons in the spinal cord. This is where, again, the big population of Answer ALS is when we make an iPS cell, your stem cell, we can convert it into a spinal motor neuron, a cortical. We have the recipes to make it into different neurons. And one of the future projects is to say, what happens when we take a patient who's bulbar onset versus, or more cortical, especially with upper motor neuron disease versus spinal? What if we make both of those set of neurons? Are there differences that we can learn? So these are incredibly powerful tools to understand the disease when given to others who want to ask those questions.
Great, thank you so much.
You bet.
And on that, so I know that there are other questions for Dr. Rothstein, and it's definitely something that we can post to him in the future or through email. I've got copies of all the chat questions and the whole chat together, so we will be able to forward on those questions to get full detailed answers and to add to the parts that were all the studies. I saw a lot of studies in the chat, so we'll make sure that those get either forwarded or added to those questions as well. We're going to kind of move to our open forum model now, where everyone is able to speak. The biggest thing that I wanted to make sure that we mentioned is that in 2 weeks, Dr. Bedlack from Duke University will be talking on our call to present his research as we continue our cure part of the webinars, and then follow up in August will all be about the care side. So we'll kind of address that side too. So in just a bit, we'll have Michelle take everyone's mute off, and we can see how everybody's doing today. Thank you, Dr. Rothstein, so much for coming and joining us. This has been wonderful, and we appreciate your time and all your expertise.
Thank you, Jeff.